Bismillahirrahmanirrahim. The alpha cells of the pancreas, they secrete glucagon hormone. The features of this hormone are discussed in this video. Glucagon is a hormone secreted by the alpha cells of the islets of the Langerhans in the pancreas, and it is released when blood glucose concentration falls. That means there is hypoglycemia. This is a large polypeptide hormone with a molecular weight of 3485, and it is a single chain of 29 amino acid. Its main function is to increase blood glucose concentration. Its function is opposite effect of the insulin. Because of this action, glucagon is called hyperglycemic hormone. The injection of just 1 microgram per kg of glucagon can increase blood glucose by about 20 milligram per 100 ml of the blood glucose. That means about 25% rise. And this effect is achieved within 20 minutes of the injection. So this is a glucose lowering the insulin that lowers the blood glucose. It has got the opposite effect of the insulin, and it increases the blood glucose level. This antagonistic relationship between insulin and glucagon is vital for maintaining a stable blood glucose level. So glycogenolysis is one of the effect or of the glucagon, that is the breakdown of liver glycogen. This glucagon activate enzyme in the liver that break down glycogen into glucose. This process rapidly increases blood glucose concentration by releasing stored glucose into the circulation. Also, there is increased gluconeogenesis. That means the glucose production from the other sources is increased. So this is the secondary effect. Glucagon stimulates liver to form glucose from non-carbohydrate sources such as amino acid, glycerol, and lactate. This is especially important during fasting or hypoglycemia to maintain the blood glucose level. So both the glycogenolysis and the gluconeogenesis, they ensure a steady glucose supply to the organ, particularly to the brain and red blood cells during times of low blood sugar. When glucagon binds to its receptor on liver cell membrane, it initiates a powerful amplification pathway. Glucagon activates adenylyl cyclase, that is an enzyme in the cell membrane, and due to this activation, it converts ATP into cyclic AMP. So there is increased concentration of cyclic AMP that activate protein kinase regulator proteins. And due to this activation, the protein kinases are activated. This protein kinase activated, and protein kinase then is responsible for activation of phosphorylase B kinase that converts phosphorylase B into phosphorylase A, that is the active form. And phosphorylase A is then responsible for breakdown of glycogen into glucose 1-phosphate. And glucose 1-phosphate is dephosphorylated, and it results in formation of glucose which is released into the blood. So this cascade amplifies the original signal. A signal a single glucagon molecule can trigger millions of glucose molecule to be released. Only 1 microgram of glucagon can double blood glucose level in minutes. So continuous glucagon infusion about 4 hours can completely deplete liver glycogen stored due to sustained glycogenolysis. So this glucagon is a potent hyperglycemic hormone because it activates rapid glucose release through an amplified cyclic AMP second messenger pathway. Even after the liver glycogen stores are fully depleted, glucagon continues to raise the blood glucose level. This is due to increased gluconeogenesis, the formation of uh, glucose from non-carbohydrate sources. And the mechanism of this is that the glucagon increases amino acid transport into the liver cell, and there is conversion of to the glucose. These amino acids are converted to glucose through the process of gluconeogenesis. Glucagon activate key enzymes involved in gluconeogenesis. Particularly, it stimulates the enzyme system converting pyruvate into into glucose, that is phosphoenol pyruvate. This pyruvate is converted into phosphoenol pyruvate, which is a rate limiting step in this process, and this phosphoenol pyruvate is then converted into glucose. So this ensures continuous glucose production even during prolonged fasting or when glycogen reserves are gone. It also helps to maintain a stable blood glucose level for organs that are heavily relied on the glucose, for example, the brain. There are some other effects of the glucagon also. There are effects on fat metabolism. At high level, it activates adipose tissue lipases. So triglycerides are converted into fatty acid. This breakdown will, along with this breakdown, will release lot of free fatty acid into the circulation, and this increase free fatty acids, they will inactivate lipoprotein lipases, and there is also prevention of the liver by the and taking up and storing the free fatty acids, and this further raises the concentration of the free fatty acids in the blood. These fatty acids are now main energy substrate except brain, where only the glucose is the fuel. So then there are some other effects of the glucagon at high level. Some are the cardiovascular effects. At high level, it enhances the cardiac contractility, or ionotropy. The strength of the heart contraction is enhanced. It also increases blood flow in some tissues, especially kidneys. At high level, it enhances bile secretion from the bile secretion and from the liver, and inhibit gastric acid secretion, reducing acidity. These effects are secondary and typically occurred only when glucagon levels are very high, such as during stress, prolonged fasting, or pharmacological doses. The primary and most vital role of glucagon remains raising the blood glucose via glycogenolysis and gluconeogenesis. Blood glucose concentration is the strongest factor controlling glucagon secretion. There is inverse relationship. Low blood glucose, that is hypoglycemia, stimulates the glucagon secretion, while the high blood glucose, that is hyperglycemia, inhibit the glucagon secretion, and its effects are opposite to the insulin. The increased glucose stimulates insulin secretion. It suppresses the glucagon secretion. Conversely, low glucose level increases glucagon secretion but reduces insulin secretion. In hypoglycemia, blood glucose less than 90 mg per 100 ml, glucagon secretion increases several fold, and this glucagon triggers the liver to release glucose via glycogenolysis, the breakdown of the glycogen, and gluconeogenesis, that is producing the glucose from other sources. This helps to correct the hypoglycemia and restore the blood glucose balance. So this graph is illustrating as the blood glucose falls below normal, glucagon secretion rises. Glucagon secretion rises steeply. As glucose rises, hyperglycemia occurs, glucagon secretion declines. That is evident from this graph. This inverse relationship between blood glucose concentration and plasma glucose concentration. On the x-axis there is blood glucose, while on the y-axis there is plasma glucagon concentration here on the y-axis. So when blood glucose is low, when blood glucose is low, 50 to 60 milligram per 100 ml, plasma glucagon concentration is high. It is about 4 times of normal, while the blood glucose concentration increases toward 90 to 100 ml mg, 90 to 100 mg per 100 ml. The plasma glucagon secretion decreases sharply. At the high glucose, 120 milligram per 100 ml of the blood, the glucagon secretion falls to below 1 time of the normal, or it touches to the normal. So what is the physiologic meaning of this graph? This is that low glucose or hypoglycemia stimulates the secretion of the glucagon to raise the blood glucose by promoting glycogenolysis and new gluconeogenesis in the liver, and high glucose concentration will suppress the release of the glucagon. So amino acids act as stimulator of glucagon. High blood amino acid level after a protein-rich meal stimulate glucagon secretion. The amino acid like alanine and arginine are particularly strong stimulator, and these glucagon and the insulin, they work together. Unlike their opposite role with glucose, both insulin and glucagon are stimulated by amino acid. Why? Because insulin promote amino acid uptake by glucose and protein synthesis, and glucagon stimulate conversion of amino acid to glucose via gluconeogenesis, ensuring a steady energy supply. So, after a protein-rich meal, glucose level may not rise significantly, and without glucagon, increased insulin could lead to hypoglycemia, that is low glucose, and glucagon prevent this by increasing glucose production from amino acid. Glucagon also increase during exercise. So exhaustive exercise can increase blood glucagon level to 4 to 5 times. This occurs even when the blood glucose concentration does not drop significantly, and the purpose of the glucagon increase is to prevent a fall in blood glucose during prolonged or intense physical activity. It maintains glucose availability for energy, especially for brain and working muscle. And possible mechanisms are increased circulating amino acids from muscle breakdown during exercise, may stimulate glucagon secretion, and beta adrenergic stimulation or sympathetic nervous system activation during exercise could also trigger glucagon release. So this rise is in the glucagon ensures that liver glycogen breakdown, that is glycogenolysis, and gluconeogenesis provide sufficient glucose to bloodstream to fuel the activity of the muscle. Thank you for watching this video. If you have any query, please write in the comment box.